Hey guys, welcome to Layla Teachers. Today we'll speak about amino acids and some reactions. So, amino acids, there are 20 of them that you need to know. It's very important for you to know the structures of these amino acids. They can come as multiple choice questions, or you'll be asked like an open question to draw them. So first we have amino acids, then they become peptides, and then they elongate to form proteins. We are mainly interested in the amino acids and peptides because proteins are too large to draw. Okay, amino acid reaction into peptides is a condensation reaction, which means that you add two molecules to get one product and you lose water. Okay, speaking about the general structure of amino acids, you have a central carbon with obviously 4 bonds. One of them is definitely hydrogen, then another one is carboxylic acid, then you have an amine group, and finally an alkyl group. In glycine, which is the simplest amino acid, instead of the alkyl group, you just have a hydrogen, so 2 hydrogens. Okay, so amino acids, because of these two groups, amine and carboxylic acids, they are known as zwitterions because they can act as both acids and bases. So the general term is amphoteric, but when you speak about amino acids, they're known as zwitterion because so the carboxylic acid can donate a hydrogen and be an acid, and the amine group can receive a hydrogen or proton and be a base. Okay, let's go through them together. So arginine, histidine, and lysine, they are the basic amino acids. Then you have two acids. You have aspartic acid and then you have glutamic acid. Okay, we have cysteine, which has a thiol group, SH. We have glycine, which has no alkyl group, an extra hydrogen, and then you have proline, which is a cyclic amine. Another amino acid is methionine. So remember, cysteine and methionine are sulfur-containing amino acids. Then you have serine with the hydroxyl group, threonine with the hydroxyl group, so they're both polar. Asparagine is the amino form of aspartic acid, and glutamine is the amino form of glutamic acid. So not a carboxylic acid at the end of the chain, but an amide instead. Okay, amino acids with hydrophobic side chains, so they are not polar. You have alanine with just a methyl group, valine, isoleucine, leucine, methionine, as I mentioned earlier, a sulfur-containing group, phenylalanine, tyrosine, and tryptophan. Two amino acids will give you a dipeptide. A dipeptide with another amino acid will give you a tripeptide, and so on. You have also oligopeptides, tetrapeptides, but I think in chemistry exams they generally just ask for dipeptides or even tripeptides. So for example, we'll take one glycine and one alanine. So draw the general structure and then put the functional group. For glycine we have just hydrogen, and for alanine we just have an extra methyl group. So a condensation reaction is similar to esterification, but instead of carboxylic acid and alcohol, like in esters, we have carboxylic acid and amines. So we lose water. We take an O and H from the carboxylic acid and one hydrogen from the amine group, and then we get what is known as a peptide bond. When you have the formation of peptides, you also have the hydrolysis, which is you break the peptide bond and you get the initial amino acid. So let's take the dipeptide that we just formed. So here you will break the peptide bond and you will attach the hydroxyl group to the carboxylic group of glycine and then the hydrogen to the amine group of alanine to get the initial amino acids, which were glycine and alanine. Now in the condensation reaction, we removed one molecule of water, so in hydrolysis we will add one molecule of water. Amino acids also undergo deamination and decarboxylation because they have the functional groups, an amine group and a carboxylic group. So Deamination is when you remove an amino group from the carboxylic acid, and decarboxylation is when you remove a carbon dioxide from the amino acid. So when you remove an amine group, you get a keto acid, and when you remove a carboxylic group, you get an amine. Why? Because when you remove the carboxylic acid on the right, you are left with just an amine group. So the whole compound becomes an amine. And when you remove the amine, then the compound will become an acid, and it is known as a keto acid. Here you can see an amino acid, then deamination, and you get the keto acid. So you have the original carboxylic acid, but then the central carbon gains an oxygen and becomes a ketone. In the second one, you can see decarboxylation. Tyrosine is an amino acid, and after decarboxylation, it becomes an amine, so tyramine, and you release carbon dioxide. Now let's move on to the homework for today. So the first question is draw L-alanine. If you remember from Fisher projection, I taught you in another video. You should be able to do this question. Number 2 is show the reaction between alanine and threonine. And that is it for this video, guys. Take care. Bye.